Thank you for coming to the training school. And for those who don't know me, I'm Silke van der Wijngaard. I'm an aquatic microbial ecologist with roots in freshwater, currently based in Finland at the University of Turku, where I'm now actually also starting to adventure in more saline waters and going to more brackish water systems like the Baltic Sea. But since my PhD, I actually have been always quite interested in these fungal parasites of phytoplankton, and also I kind of like got a like of trying to isolate and cultivate them. So and I think that's also why I'm now giving this kind of talk about like how can we isolate and cultivate these parasites, because I think we are all kind of convinced that this is kind of an interesting thing to do and a valuable thing to do if we want also to know more about like how actually these parasites cause disease, which is then more the topic of tomorrow. So what I here would like to do is just share my personal, mainly my personal experiences and tips and tricks of how I kind of approach this. And yeah, let's start. So with the basic outline. So the target groups that I will kind of like specify and focus on will be parasitic chytrids, so fungal parasites of phytoplankton. So just to be aware, it's not about all parasites, but it's quite specifically tailored for this group. How to become a chytrid isolation star. And then we go to the practical steps, going from sampling, screening, detection, incubation, isolation to cultivation. And then a few words on morphological and molecular characterization of those parasites that we then isolated. And again, this will be very highly personal, biased talk. So, but I hope that maybe also some of those general concepts or methods can also be applied to other zoosporic parasites or other host associations. So maybe from like going back to tomorrow, where the focus was more on not having the contaminants, here actually we are wanting to attract the contaminants, the parasites. That's our goal. But just to give, of course, a very brief, before we are jumping to this very specified group of fungal parasites, just that you are aware that this is, of course, not the only group of zoosporic parasites that are infecting phytoplankton or microalgae in general. So we have a very diverse groups of these zoosporic parasites, which you can find in many different geographic lineages. So like in Rhizarias, Thermonophiles, alveolates, just that you are aware that we are now looking at just one group of those zoosporic fungal parasites. So our targets are, again, like this, the chytrids, fungal parasites. And just some basic main facts. We can find them in both freshwater and marine systems, so everywhere, also natural, artificial systems. So everywhere actually we are going, we have the potential to isolate them. They are infecting all major phytoplankton taxa. And some characteristics are that they have this uniflagellated zoospore, so that's how we also kind of differentiate them from other zoosporic parasites, which are usually biflagellates. So this fungal group has just a single flagella. And then they also have this mainly epibiotic thallus. So we see that the vegetative structure develops outside, like on the surface of the host cell, and the infections are lethal. So, and very briefly, also a bit an overview of the life cycle. And many of you probably already know. We have, of course, a free living zoospore, infective motile stage, that's swimming in the environment to find the host, and then it attaches and insists on the host surface. It develops rhizoidal structures, which you don't really see now in this picture, but with this rhizoidal structure, it penetrates the host cell and absorbs nutrients from the host, and it produces new infective propagules that are released then again in the environment. And when we are isolating, we are targeting those host-attached vegetative stages. So just to point out that. Okay, how now become a characterization star? So I think we are all aware that having those cultures is really valuable. And this is also what is expressed often like in papers. So they are, of course, very good if you want to do species descriptions or if you want to do experiments. You need often those culture systems. If you want to understand more about like how those interactions work. But then you get always this kind of very demotivated messages like, this is difficult. It's extremely difficult thing to do. And I see that I'm also actually guilty of some of those really depressing messages. So I want to make that up. And I think this is a more realistic quote that we can use. So yes, maybe it's not an easy task, that's for sure, but probably it's also because little effort has actually been devoted to this task. So my message here would be more like, this is not impossible, everyone is capable to do this. So we can start with this motivating sentence, we can continue. So what are the skills that I think when I was thinking about like what would be the mindset of starting with isolating parasites? It's I just here gave an analog of MacGyver. It's not necessary that you know this person, but the skills are, I think, in my opinion, observation is very important. So you have to have an observation skill, improvisation, and be flexible, creative is helpful. Very important is also patience and perseverance, and then also a difficult one, discipline. So let's have, with this mindset, I think we are also ready to go, to start. Also very important, and this is actually the basis and skills in microalgae isolation and cultivation, because in the end, we are isolating parasites on algae. So you first need to have your algae before you can isolate the parasite. And I'm really very happy that Anna already gave in the morning like the about this, about microalgae cultivation. So this is really, how you say, the basis of starting to build up on parasite isolation. Some basic skills in light, fluorescence microscopy is also useful. And very, very, very helpful is having a great mentor. And I put this picture and I hope, Elena, you're not, it's okay for you. But so in my case, my mentor was 15 years ago when I started my PhD, Alena Gesell, who is now here also in the training school, who will be teaching tomorrow. She introduced me to really the basic skills of microalgae cultivation and parasite isolation. So that was for me, and I think now for you, hopefully this will be then this training school where you have really great mentors and teachers like Anna, Gabriel, Yurika, Dagmar, and Kayo to get you into it. Because this is really very helpful, because this theory is just theory, but it's really the practical things that then are really the most important things that to get this going. So let's start with the first step, the sampling. So when do you want to go out for isolating parasites? And of course, this is system dependent. I mean, in cultivation systems, I mean, there are the reactors, so you can basically go probably at any time point. But if you work in natural systems and in temperate regions, you have seasonality. So that means that you have different phytoplankton species that are blooming and occurring at different time points. So it depends very much on what kind of parasites you want to isolate from which host species. In principle, you can actually find them all year round. So this has been demonstrated in several papers looking in lakes, in temperate regions. But of course, if you are interested in isolating parasites of diatoms, then the diatom spring bloom is a perfect period to go and catch your sample. So just as a general rule, the best time period is when your target host species are the most abundant. And then how do we sample? That's also, of course, system dependent. You want to have sufficient amount of material, not that you have to screen in the microscope like half an hour to find a cell. In cultivation systems again, or in very eutrophied lakes, probably just a scoop of water is enough. If you're going to natural environments with a bit of lower nutrient status, which have relatively low cell densities, then you would like to have this a bit of a concentration, cell concentration or enrichment step. And we use a plankton net, for example, for that 10 micrometer plankton net. So this ensures that you collect all organisms bigger as 10 micrometer, which most of the microalgae that are potential hosts are in those size ranges, so 10 micrometer or more. And then you just concentrate them with the phytoplankton net. This is another workflow just took from last week, where we were on the Baltic Sea, where we are actually on a ship, and we take we take actually the sample via tap. So the ship is going, like, it's moving, and we are collecting the water in these canisters and then concentrating it like this through the plankton nets. And then also what you might consider is to remove potential predators from your cells, so to avoid that when you are having then your sample like back to the lab, that all the cells are not eaten by zooplankton that are in there, like the phytoplankton, but also the parasites can be susceptible to grazing. So usually we sieve the concentrated cell suspension over 100 or 250 micron mesh to remove those predators. And then it's maybe a bit difficult to see. This is kind of like how then a sample could look like. It's maybe not really visible, but there are flocks inside. So this is a kind of an enriched sample that you then will take to the lab and to further analyze. So at the screening step, what is some kind of basic microscopy equipment that we need is definitely an inverted light microscope. I would say with 10, 20, 40, and potentially also 60 objectives, but 20 and 40 objectives are usually the mostly used ones. And then some recommended options are if you have like objectives with phase contrast or differential interference contrast to see like better structures, fluorescence, and a microscopic camera, or nowadays, of course, you can also just use a smartphone, so it's not really so mandatory anymore. And then microscopy chambers with your samples in. So we often use, like mostly use these Utermold chambers, which are very much used in phytoplankton research. But of course, you can also use Petri dishes or well plates, but preferable something with the glass cover slip bottom, because then you have like a better optical resolution. You have to just take into account that when you're working with plastic, like thicker plastic, then you will have a lower optical resolution, so recognition of structures will be a bit more difficult. And then you would definitely need long working distance objectives. So that's something to keep in mind. Then what are the criteria to diagnose those parasitic hybrids? So when we are looking into this sample, what do we have to pay attention to, and how can we identify those parasite structures? So first of all, we look at these outline globose to oval shaped structures that are physically attached to a phytoplankton cell. Then the size of that attached structure or sporangium should not largely exceed the host cell size. Another criteria that you can use is if the phytoplankton cell that carries that structure shows signs of chloroplast degradation. So that may be an indication that there is a parasitic interaction going on. Then if you have fluorescence, you can use autofluorescence for that. But of course, if you are looking at lugal preserved samples, then that autofluorescence will not work. If you also, again, if you are looking at with fluorescence microscope, you can also check if that attached structure has autofluorescence or not. So parasitic fungi, they don't have autofluorescence. That's also a criteria. And then if you look into the sample and you see the reoccurrence of sporangia at different development stages on that same phytoplankton species, that can also give you an indication that there is a parasite involved. And here's some examples. So in the left picture here, why do we don't consider this as a parasitic fungi? First of all, the size of the sporangia structure is really very big compared to the host cell size. And then also the associated host cell do not show any signs of degradation, even if that sporangia is actually kind of major and mature. So that gives me something that says like, no, this is not a fungal parasite. On the other hand, on the right side, we have realistic shape and size. And we also see that the associated host cell is kind of depleted. So this is a very good candidate. Another one here on the left side, it's very difficult to see maybe here, but here we can somehow detect a kind of flagella. So sporangia structures don't have any flagella. So, and also again here the host cells is kind of healthy, looks healthy. There is no signs of degradation, even if it kind of is completely colonized by something, but it's definitely not then a fungal parasite. On the other hand, on the right-hand side, we have really a nice example of a fungal parasite again. So again, such kind of realistic shape and size, and the host cell, which is kind of largely degraded. And I mentioned that having fluorescence can help, like to then, because then if you have a fluorescence microscope, you can use stains, and for example, you can use fluorescent chitin stains that can aid in detecting those fungal parasites because of the fungal cell wall, which has chitin. So there are two stains which are commonly used, that is chalcofluor white and bleacher agglutinin conjugated with the fluorochrome. I just here noted down like the filters of the microscope that you would need to use those dyes for your information. And also I would not want to go like too deeply now in this fluorescent staining methods. This was like part of the first training school. There was a lecture on that, and I can recommend this paper that really provides a nice guidelines for using those different dyes on fungal parasites. So if you want to read a bit more on that, you can go to that paper. As an example of how we can use the fluorescence to detect fungal parasites, on the left, as I mentioned, fungal parasites don't have autofluorescence. So if we see this kind of structure, which would theoretically be kind of in the size, like theoretically a good size of a parasite attached to a spore, but it has autofluorescence, so that eliminates it from being a parasite. On the other hand, here we have similar size, and we have a positive stain with the chitin stain. And even here you can see the rhizoidal structure inside. So that's a positive parasite. So just as an example of how this actually also can aid in diagnosing a parasite infection or not. Then when we are screening the sample, important is we want to collect all relevant information. So observe the sample, make notes, drawings, take pictures. It's very useful also for afterwards. And just very basic questions like how does community composition looks like, what are the most abundant species, which species of course are infected, what is the infection prevalence, Are there different parasites, morphotypes infecting the same species? Everything, what would you kind of like observe and which may be of relevance, you just note it down. And then after that first screening, we have to make a plan, like how to continue for the isolation. So the most important questions, of course, were, are there high-grade infected phytoplankton cells in the sample already visible? And how high is that infection prevalence? Is that already an epidemic stage, or do we only see just a very few amount of cells? And if we see infections, like do we actually have a culture of that host species that we saw in that sample that is infected? Because that will kind of determine a bit like how we proceed. If we now have a very high epidemic of a phytoplankton species, like the species is highly infected, and we have that culture, we can go immediately right away to the isolation phase. But if we don't see any infections yet, or just very few, or we don't have the culture yet, then we want to maybe incubate or like enrich those parasites. So our goal is then that we want to have, of course, as many high-grade parasites to isolate. And that means that we have to provide the conditions that promote parasite development. And in essence, it means that we have to make it easy for the parasite to transmit and reproduce in our sample, to increase the amount of infections, to isolate. So then the very difficult question, what are the optimal conditions, drivers that promote high-grade parasites? Well, this is usually something that we actually don't know and which will be the topic of tomorrow. But maybe something very simple to consider is, of course, like what promotes high-grade parasites is something that limits parasite loss. And what limits parasite loss? We already have removed the potential predators. If you did not already did it directly after the sampling, this is something definitely to do to avoid that any predators are eating up your parasites and interfering with the transmission. Then you want to promote an abundant potential host population to facilitate parasite transmission. And for this, you can either add the culture strain of the host species that you have already, like if you have it already in culture, you add it, so ideally the same host species or something, like species that is closely related to the species that is infected. Or if you don't have the host culture already available, you can add appropriate culture medium, so microalgae culture medium, to the sample in order to stimulate the growth of the target host species. And in double fold, so you want to try to isolate it, while in the meantime maintaining and keeping infections alive on that host in the natural sample. It's a bit tricky, of course, so like it is more difficult if you don't have the host culture. So the recommendation is, of course, always like to, if you know which parasite species or which host you want to isolate, is first to get the host into culture before you are going to try to isolate the parasite. But it's not impossible to do it in this way either. And then, of course, depending on the time and resources, you can try several things. You can play around with different abiotic conditions or propose different strains of host species if you have different strains of the same species. But very important then is when you are having then this incubation running, then you have to just screen your samples frequently, daily, every second day, and to be ready when something happens to take action. Because then if something has happened, so we have the infections going on, we can move on to the isolation step. And isolation step, like in what I want to say with this is then we want to manually pick a single infected cell and put that on a pure host culture. So I'm using a very, maybe simple technique is just a micropipette, a 10 microliter micropipette. And that works quite well for me. I know that there are other techniques of isolating single cells. But this is a very simple one. So you just need a micropipette, autoclave tips, and again, kind of a microscopic chamber where you put your sample in. And then the method of is very simple as pipetting just normal liquid fluid. So before you entering the water sample, you just press the pipette to the first stop, you enter the sample, and then the most difficult part comes then, because then you have to somehow focus the tip of the pipette. So with the oculars, you have to somehow see then, like, and get the focus of that tip on the bottom of the microscopic chamber to pick up the cell. And this is something you just have to do by practice. So this is something that you just not like, it's just impossible to just tell stories about it. It's just practice. And I hope that you will be able to practice this kind of single cell picking then in the practical part. Yeah, also a maybe very trivial thing, but so the tip of such a micropipette is still, of course, quite large. So it's around 300 micrometer. And what is then, of course, important is that in your sample, you're having not a too crowded sample where it's just impossible to pick up a single cell. So this is of course, way too crowded if you would here try to pick a single cell, which must be impossible. So ideally, you want to have something like this in your vision that, yeah, you can pick up only a single cell. Well, you will never pick up the first time a single cell, but that's not a problem. So we have washing steps in between. So here, for the washing steps, we transfer then that picked single cell in drops of cultivation medium. So just the cultivation medium that you are using for cultivating your microalgal host. We are using just clean Petri dishes for that, or you can also just use a thermal chamber, whatever. But the goal of those washing steps is then, of course, to remove contaminants, and the definition here, what we have of contaminants is then just other eukaryotic cells or any kind of debris. It will be impossible to avoid bacteria contamination. So this technique is not for establishing actinic cultures. So that's not a goal that we have, but it is mainly for removing all other eukaryotic contaminants. And of course, we want to ensure that we have a single pair, that means a single host cell with a single sporangium in our final drop, so that we have we can establish monoclonal cultures. And usually, kind of a rule of thumb is that we have needed these 3 washing transfers to get to this kind of final single cell. But of course, you have to kind of screen your final drop if you're really sure you have nothing else in there. And if that is the case, then you can transfer it to a host cell suspension. And this we usually do, we take these 24 well plates that we prepare in advance. We fill wells with 1.5 milliliter of host cell suspension. Ideally, you use exponentially growing host culture. And when considering host cell density, there is you want to have, of course, a high enough density that allows parasite transmission, but you also want to have it low enough that it still allows a few rounds of host cell division, so that you still have some activity of the host cells before they are reaching into a decline phase. So you have to find a bit of that balance. Optional, of course, if you have many infected cells available, you have many trials that you can do, then you can just try, of course, with different host strains if you have them available, because we know that sometimes parasites are even host strain specific, so you might increase the chances of getting a successful culture if you are increasing the number of host strains, and trying with different cell densities. That also can increase the chances of getting a successful infection. So isolation so now we have our single infected cell. We have put them in different wells, like with different host cell suspensions. So now it's time to follow those up and to screen for new infected host cells to see if the parasite successfully can transmit to new host cells. And for this, yeah, it's again the same, just frequently observe. I would say daily, every second day, have a look what happens in all of the wells and mark those with potential infections. And from experience, usually it takes at least around 5-7 days before you actually can see new infections in those wells. But the skill of patience comes here back again. Be patient. Sometimes it really can take a bit longer, 2 to 3 weeks. But I would say if after 1 month you really did not see anything happening yet, then maybe it's time to move on and to discard the well plate. Then we are, so in the good case, we have in a well, we have something that successfully like transmitted. So we have something, a parasite that is successfully growing onto a host culture. And then we want to scale that up and we want to establish a stable co-culture of that. So when you have this kind of prevalence of infection, so if the proportion of infected cells is around 50 to 75% in that well, then it's kind of ready to be transferred. And then I usually take like 1 milliliter of that infected cell suspension, and I put it on a new and healthy host culture. Again, like preferably exponential or early stationary growth phase. And yeah, then it's waiting and it's seeing like how it behaves, because it's not always it's not always that it goes very smooth into a predictable infection dynamic. So this is a kind of like the phase now that you want to find out what are the conditions that I can kind of reliably have this stable infection dynamic, that I know, okay, when I put that and that together after so many times, I need to transfer again. So you want to get to those kind of routine conditions. And this demands a bit of fine-tuning in the beginning with what is the starting host and parasite densities for the transfer. And very important in this process is that you at least know how your growth, how your host is behaving. So how the growth curve is of your host, because that gives you then information already about like how long is exponential phase of your host culture under, of course, certain environmental conditions. Because looking here, I can just plot it here, kind of like an ideal a potential infection dynamic, which I have with my cultures. So I usually start with the host density, and that allows kind of like still host growth in the beginning. So you don't want immediately that your parasite, so you start with a very low prevalence of infection when you transfer, because you don't want, of course, that from the beginning your parasite is taking over and infecting everything in 2 days. So you still want to allow your host to have a little bit of a time window to increase in abundance. And then, of course, the parasite will take more and more over. It will exponentially grow and it will start to infect and decline the host culture until you come here in this phase where you have almost no healthy hosts anymore, but you have a high amount of parasites. And of course, you also want, if this is a kind of the time of transfer then, because you, of course, you don't want to wait until all your hosts are gone and all your parasites are gone. So this is the time window, and then you have to be there for transferring the parasite again to those new conditions. And So this is now a scheme that works for me, and I have with my cultures that I have this common transfer frequency every 7-12 days at incubation temperatures of 16 to 80 degrees. So of course the temperature determines how fast your host and parasite grows. So of course you can slow processes down when you reduce the temperature. And then theoretically you can also slow down the frequency of transfer. But I only recommend that to do that when you really have a stable co-culture, where you know, like you have predictable dynamics, and then you can start to somehow play around with changing the environmental conditions. And also very important, that temperature is really an important factor, of course, for both host and parasite. And what we have seen from experience a lot is that commonly diatom-associated hybrids, they are really quite sensitive to high temperatures. So temperatures above 22 degrees can become critical for the parasite. Just something to also keep in mind. So what is now the secret to successfully maintain a phytoplankton hybrid co-culture? For me, I think is the most important is that you are able to maintain a healthy and good growing phytoplankton host culture. So usually from experience, when parasite cultures are crashing, it's not really because of the parasite, it's because of the host. Because of the host is not doing well. And as we are here often isolating obligate parasites, so parasites that are really need and actively like living host cell, the health of the phytoplankton host is really crucial. So that is really the most important thing that's like that keeps it the success or let it fail. Then of course being able to maintain a controlled abiotic environment, so having growth chambers will help to stabilize conditions. Frequent observation. This is when establishing the culture, for sure. You really need to be very actively involved, and like if something strange is happening, just to kind of interfere and try out things to. So here you really have to be reactive. And also even when you have a stable cultivation, I usually just minimum check once a week when it's time to transfer, like just quickly the cultures. But that's just like some discipline that you need. You just have to put some minimum of time investment in it. And having, of course, multiple backups of both the host cultures and host hybrid co-cultures, that helps a lot in time and in space. Here, well, we don't have it so far. We like these culture collections would be, of course, something very promising, like to have that availability more, like to as a backup also for those systems. And maybe also like this cryopreservation protocol development. So there has been some work on that, but not so much. So I think that would also still be interesting to develop that more, that one can kind of cryopreserve something, and that you don't need this kind of very time-consuming steps of keeping the cultures active. So then we are going to morphological characterization. A few words on that. So because when we have the culture, of course, so now we have that stable co-culture, we can start to observe all those development stages of the parasite life cycle. And of course, again, this will then also allow to do experiments to investigate drivers. And what are the characters that we are like mainly looking at for morphological characterization? Is like, first we have the zoospores. Well, there are not so much of like really you say distinct traits in zoospores. There can be some variation in shape and body size, but usually, commonly, they are quite just small and spherical. And they have a single lipid globule, although also some species have multiple, but the common kind of like most of the species of the hybrid species are quite similar in shape and have this single lipid globule. But of course, it can be a criteria for species differentiation. Then the talus, like sporangium features. Here we look at shape and size of the major talus. So this can be kind of also variable. We have spherical, subspherical sporangia, more ovoid to pear-shaped ones. So this is also something, how you say, a trait, like a characteristic that you can use if you want to identify a species. But be aware of always that the shape can be also dependent on the host species it infects. So it's not always a fixed trait. And also the size, of course, it's very dependent on, can be dependent on the host cell size or on environmental conditions. So it's not a fixed trait, it's a flexible trait. Another interesting feature is the infection site. So some species really have preferred infection sites on here on the ends of filaments, whereas others are infecting in the middle, or here on these desmids, some species are infecting on like any side of the host cell surface, but some are really only infecting here within the central region. So that is something you can notice in some cultures. And then the zoospore discharge structures, those can be really quite variable and quite distinct between species. So we have two major groups, like the operculate, which are having this kind of little lid, operculum, that kind of opens up and then zoospores are coming out. And then you have the inoperculate ones, so without a lid, so where there is just kind of like a pore where zoospores are moving out. And those inoperculate ones, they can be different. Like you can have species which have just a single discharge pore, or you can have species where there are multiple discharge pores. And they can even be species which actually you don't even see any discharge pore because the cell wall just completely dissolves. So in the end, you will not kind of see any rests of empty sporangia because it's just dissolves completely. So that are traits. Rhizoid structures. So here, these are actually not always very prominent and very, very easy to see. But here again, chitin stains can help in visualizing those a bit better in some cases. So here the traits are that you can have unbranched or branched. So here, for example, you have a short unbranched rhizoid, whereas in this case you have kind of like a short branched one. And here you really see a very highly branched rhizoid going through the whole cell is quite thick. So these are also kind of different traits, like characteristics, which are also species dependent, but might also be host dependent. And that's always something to take into account. Then the resting spores. These are characterized by thick cell walls, so you recognize them when they have like these very thick cell walls, and they can be asexually or sexually formed. So here is asexual, but here in the middle you see this kind of like donor cell. So this results from like two kind of thalli, and then one is the donor and this is the recipient. This forms then a zygote. So those are formed to escape unfavorable conditions. For example, when there is like the host is absent or gets in low abundance, then parasites might develop these resting spores. Not all hybrid species may produce them, or at least not under culture conditions. So in culture conditions, it's not always that those resting stages are formed, but it can happen. And then of course, usually it's at the end of the infection phase when all cells are infected and nothing, like no healthy hosts are available anymore. Some other additional features are this kind of like funny, like stalks. So some are sessile, but then some of the species form these little stalks to attach to host cells. But in general, so this kind of like collecting all those morphological traits, if we do the example of the cell, like the single cell that we isolated, Staurastrum on this desmid species, we can kind of make a summary of the features. So in our case, you would say, okay, we have a host species, is Staurastrum, maybe others, dependent if you manage to isolate it or on other species as well. Then we have the zoospore, let's say it's a normal zoospore, spherical, 3 micrometer. The sporanium is sessile and it's spherical. The infection site seems to be variable, so we have both here in central regions, but also all over the surface. Then we have these multiple discharge pores that we have observed, and the cell wall is persistent. Rhizoids, we are not really be able to identify, to see them at least, so maybe they are not prominent, or maybe we can just not visualize them properly here. And we did not see any resting spores. So with that information, then our question, does this parasite resembles then a species that has been observed previously and has been described previously by others? And where to start? That's well, of course you can Google. That's definitely what I also do. But I have actually also 2 literature resources which I often go through too when having this kind of first basic knowledge, like the observations. Then there is a really great book, Aquatic Phycomycetes of Sparrow, which is a compilation of really a lot of zoosporic parasite species. And of course, there is a whole literature of Hilda Counter. She has more than 70 publications on phytoplankton parasites, mainly chytrids. So I usually kind of like go to that literature. And the first thing, of course, is to see like what are hybrid species that have been described from the same host species, like to start with. So in the Aquatic Phycomycete book, you have this kind of list with host species and then the different parasite species that has been described. So you have already some indications what species are infecting Staurastrum. And here, for example, with the literature of Hilda Counter, so this is an interesting one where parasitism of planktonic desmid. So that's something you want to go through to see what has been described. And I'm not going to go into details in this because this is just detective work and reading work and comparing and like what do they describe and how it corresponds to what we observed. It can be quite a detective work to really like go to all that literature, but usually it's also not really very conclusive. So there, in this case, I just highlighted green things that kind of corresponds to our morphology, and in red things that do not correspond. So I have kind of 2 candidates: Rhizophidium globosum, Rhizophidium cauchi. You can also look then, what I usually do, I go to GeneBank also to see if, for example, one of the species has sequence, because of course the next step that we can do is also to molecularly characterize the species to do a sequencing of one of the rDNA genes. So that's the next step, because only based on morphology we will not in any case be able to really pinpoint species. So very briefly, molecular characterization. Collecting the material, we have the co-culture, and we usually then, of course, want to harvest that co-culture material at the end of an infection where you have the highest amount of parasites and a low amount of host cells. Usually if you have a dense culture, 1 or 2 milliliter is already enough, and you just spin it down to form a pellet. If you have the pellet, then some of like very easy and rapid and very cheap DNA extraction methods are available if you just want to extract PCR quality DNA. So in our case, when I'm talking about like basic molecular characterization, we are going for a PCR and for a Sanger sequencing approach. So then those very fast methods are at least in our cases, has been very successful. I'm not going to go to the details like of the protocol, but just list it here for your information. And what you really just only need is very basic equipment, and yeah, very easy to do. Then the primers, those are so, I then recommend to go here on the website of the Unite. They have a collection of many different fungal specific primers, because of course here we are now collecting a co-culture. So of course the advantage is that we are not, in theory we could also separate the parasites from the host if we filter out only the zoospores, for example. But if we are doing these PCRs on a co-culture, if we are using fungal specific primers, which in any case we want to do because we know we are working with hybrids here, then this co-culture like will not interfere with the PCR because we are using that fungal specific primer. So here are some of my favorite primers, which usually work very nice. You can look on the Unite website. You have all the information, the sequence, and the references to these primers. So, but those ones are usually the ones that we are using for basic molecular characterization. What you could also, of course, do is a long-range PCR. So, for example, here, this would be just kind of short stretches of ITS, LSU, and SSU. But you could also amplify kind of like the whole rRNA operon. So there have been some very nice primers designed for this, and this one short, this RCA95M by Christian Wurzbacher. And with this, you kind of like amplify the whole region, and then you could in principle do a nanopore or biosequencing of the whole stretch. But if you do a Sanger sequencing approach, you can then also, with that first PCR of the long range, you can do this kind of second internal PCRs to cover kind of like the whole RNA operon. So that's also an approach, and you find that one here in Kagami et al. And then if you have your PCR products, you can, of course, send them to your favorite sequence company. And yeah, if everything works out, then you get your raw sequences back. And then you want to just kind of look at them and edit them a bit. So here again, I use BioEdit. It's a free software you can download. It's quite easy to use, so I like it. And here you can just easily visualize then the chromatogram of a Sanger sequence, and then edit. So that means you have to just trim bad quality sequences, usually at the beginning, at the end of the sequence. And just to give an example, so this is a very nice quality chromatogram. So every peak corresponds to a base, and you have really a nice single peak. For example, if you have something like this, then you can just throw it away. So this will not work. You have multiple peaks. You don't know what your base is here. So that is something to discard. Then we have our forward sequence and our reverse sequence. So we want to align those to form a consensus sequence. And basically, when we have then that consensus sequence, we can save it as a FASTA file. So it will look something like this. You can just open it in a normal text editor. And this is kind of like your final sequence then that you can use for searching against the NCBI database to see if those that sequence is somehow corresponds to a species or an isolate in the database. So here you go just to the BLAST like websites, you use nucleotide BLAST, you copy paste here your sequence, or you uploaded it, the file, whatever, you can choose. And here I usually just mark exclude uncultured or environmental sample sequences. So that gives you only hits with like species and isolates and cultures. And yeah, then we basically, these are like the things, only things that you need to take care of, and then you hit blast and you see what comes out. So these are the hits. And what can we see then from the results? Well, we have here 89% sequence similarity with Rhizophidialis species isolates, and with Entophlictis, and here also with Paranomyces, 86%. So what it basically tells us is that the sequence of the species that we have isolated is not in GeneBank. So we know that it's not going back to the morphological characterization where we were thinking, it's Rhizophidium globosum. Well, it's not, because Rhizophidium globosum is in the database and it's definitely not here in the list. So that is kind of excluded. So we can still think about if this is Rhizophidium cauchi, but unfortunately Rhizophidium cauchi is not in the database, so we cannot verify that with our sequence. Or we have a novel species, and this is usually kind of like the most common case, like of going to this morphological and molecular characterization, is kind of these kind of conclusions that maybe it's something that we know, there are some similarities, but also some dissimilarities, so maybe it's novel. So that's what is the usual conclusion. When we just go, we click on this Rhizophidialis isolate, just by curiosity, like what information is given here. Then it's kind of interesting why is here isolation source is an heat-treated Rhizoclonium. So in this sense, this species, the most close hit to say seems to be also associated with an algae, but maybe not in a parasitic interaction, but at least something saprophytic. Yeah, so here are we again, Rhizophidium cauchi, the known unknown, or novel species, the unknown unknown. We don't have to answer that question now. Not at all. We are at the end. And I just kind of hope at least I could motivate or like people to go into this isolation and cultivation of parasites. So again, even if it's maybe not the most easy, it's not impossible, definitely not. And it's really quite fun if you get into it. And because definitely we need more people that are doing this. We need your help for isolating and sequencing what we see, all these unknowns or known unknowns. That is really something we would also like in the cost action to get more action on that. So I really thank you for listening, and questions are very welcome. So I maybe stop share the screen. Is that okay? Or
yes?
Yeah, shall I stop sharing?
Yeah. And if there are questions, please, now is the moment for Silke. Yes, please. Silke, you will tell me if you hear, okay?
Yeah.
Yeah. You mentioned how important it was to have a mentor who sort of knew the system before you started. How possible would it be for one person to just sort of decide to start this system in their own lab somewhere? Like, would you need sort of training? Would you need mentor, like you said, or could you just sort of go for it from day one on your own?
That's difficult. I would say at least for this practical isolation, like this, if you really are like want to do this microscopy like base technique and manual isolating of the cells, this is really something that I have realized that this was really I profited a lot from having it at least one time showed. I don't think it's impossible to do it yourself, like but it might take just a bit longer to get it. But I don't think it's impossible to just start off your own. It just makes it easier and probably faster for you if you have somebody showing you for the first time the steps. I think this is general, like always a bit the case.
Okay. Anybody else here? If no one, Serena has a question, I think.
Yeah, thank you. Thank you, Silke, for sharing your experience. It was really interesting. And I would like to ask you, when you cultivate then some host with parasite on the long term, if it happens to you to see some host resistance to parasite developing, and what would be some trick to go over that?
Mm-hmm. Actually, so far I lost a few cultures, but I would not say that it was due to host resistance, but it was mainly due to the thing that I was telling about, like an host culture which then just crashed, and you're providing not the suitable host. So it's not really about resistance. So far, no, I don't have had that experience that I lost a culture due to host resistance. Yeah, interesting wise. But you never say no.
Okay. Laura?
Yeah, Laura, I can, yeah, please, I can give you the word.
Maybe this is a very stupid question because I have never worked with kids, but have you ever tried to use some mimic media to try to perform some acrylic culture? Like, I don't know, some algal, smashed algal media sterilized, or some growth that can make the condition inside the host of the cell, or if some of these chytrids have some facultative stage that can be used for acrylic culture.
So I'm just going to repeat because I don't know if I understand did I understand it all correctly? So you asked about if there is some to get actinic cultures?
With some mimic media, for example, like algal, sterilized algal growth, or nutrients. I don't know. I really don't know how about chytrids or what they really need the cell inside which they grow.
Maybe I think that Laura, please correct me if I'm not right. Laura wants to ask if it's possible to culture parasitic chytrids without algae.
Yeah.
Okay. Now I understand. Sorry. Well, I never tried, so it's something to try. In the sense, I mean, you could theoretically, think about if you know what the algae are using as food, you might be able to do it. But so far, I must say, I never tried, so I cannot always also say if it works or not. The thing is that if we are using, for example, crushed algae, because this is, for example, when you want to kind of like get an idea if your parasite is really needing kind of a living host, or if it's grows on just yeah, organic matter, like that, like host organic matter, then one test is that you kind of heat kill algae and then you offer that. And then, well, in many cases, like working with these obligate parasites, it does not work. In some cases, if you have a kind of a facultative parasite, but then that's another kind of interaction. So these facultative parasites are then those that can have a life cycle on both, like living and dead. So those ones you can probably more easily cultivate on those yeah, like organic substances. But yeah, those obligate parasites seem to be somehow need something in a queue of an active like host that's so for those ones, no, I would not. So far, I think I think it's not impossible, but I would not know like where we need to start with it.
Okay, thank you. Okay, Maya. Maya also have a question.
Did I understand correctly that you're able to regulate, this relationship of algae and chytrids in your cultures in order to have a continuous culture? Because this is very interesting also for the larger scale, and for new products. So we would really like to see more of this knowledge.
Yeah.
So I understood correctly, right?
Yeah, I mean this continuous culturing, of course. I mean it's not like, it's not really continuous culturing, right? I mean you always kind of like transfer. So you always, you have your kind of host growing and you have your co-culture, and then you kind of always need to transfer newly, inoculate newly a new host culture with the parasite to maintain it. So I can imagine that, I mean, setting up a continuous culture is also possible, like to do, if you kind of like automatically provide new host cells, and then you can keep the parasite alive. It's not something that we are doing like here in our lab, so we have just batch cultures. But it's something I think that you could also try to set up. It would be interesting to have a really I would say like kind of a reactor and keep it going in there. Would actually be interesting to set up.
It have to be continuous. Just as you understand this relationship, you can manage it, because, for example, a big problem in algae is how to break the cells. And if you could do it already in the culture, maybe this would be a nice thing, but not at the same time, crash everything and,
Yeah.
Regulation is very
balance. Mm-hmm.
So there's the catch.
When to stop.
Okay, Oliver.
I know you mentioned the temperature, but did you maybe investigate like what if changes in the other parameters affect the rate of infection or the interaction between the chytrids and the algae?
So can I repeat? So the temperature, the temperature is a factor that affects. But do you ask like what are other?
And we investigated something apart from the temperature. So I don't know, salinity, pH.
Well, we are working on salinity right now, but work in progress. I think this is. I mean, Alina can probably tell you much more about that tomorrow in the in the lectures. So this will be the topic of tomorrow. So I don't going to. I mean, and I don't want to spoil, and I also have not. I will say. the best knowledge. So in a sense, when I was talking about temperature, and this is actually based on those experiments that Alena have been doing also with temperature, for example, on diatoms. So there is clearly an effect on temperature, not only due to the host growth, and like it's an interaction between host growth and parasite growth, of course, but also that temperature can really maybe also affect negatively affect the parasites. So it seems to be a bit like the tolerance ranges of the parasites seem to be a bit more narrower than the host often, but maybe not in all cases, but it seems to be the case also for temperature. So usually we can grow algal hosts at much larger temperature ranges than the parasites. And that is of course interesting for if you want to look at controlling infections. If you can change, if you can fine-tune those abiotic conditions where you know that you can still have a good growth of the host, but you kind of like almost can eliminate the parasite. That would be an ideal situation. So that's why I think, yeah, again, tomorrow's topic will give much more insights on that on that part. So this was more about, okay, this is we are now delivering the culture to Alena, and she will now take it further.
Until tomorrow, I think that Oliver is satisfied with the answer. Okay, thank you, Silke. Thank you very much.
Yeah, thank you all for listening. Bye bye.